Hello, AP Biology. This is going to be part one and what will likely be two parts of a video series for chapter 3. Chapter 3 is a pretty lengthy chapter, so I imagine we'll have to do two separate videos for it. Okay, chapter 3 is called Carbon and the Molecular Diversity of Life. And as is our sort of tradition, we're going to begin with a photograph. This is a yearbook picture from, gosh, I think 1986. Maybe 87. This is a first grade class from Chapin Elementary School in Chapin, South Carolina. Yours truly is in this picture. If you want to take just a second, see if you can pick out what first grade Mr. Kuhn looked like while I'm talking. So this is this is showing a bunch of molecular diversity. These are a bunch of organic beings, first graders, 6, 7-year-olds. I picked this picture because all of the molecules that your body is based upon, the molecules that your body constructs are based upon the element carbon. Molecules that contain carbon are called organic molecules. In college, you might have a class called organic chemistry, which is a very challenging class, but it's the chemistry of carbon, which is basically the chemistry of life. Okay, so any guesses? I know you can't answer me right now, but as to which one of these lovely people is me, I will tell you this is not the first page of the yearbook. So like this young lady is not last name beginning with A. This is in the middle. I'll just tell you this one right here, this cutie pie right here, that's me. I love the haircut too, right? Nice bowl cut. Maybe I should sport that again. Okay, so let's just get started. So we've already kind of said what's on this slide. So organic compounds are carbons that contain, are molecules that contain carbon. The last point on the slide uses the term molecular architecture. So your body is basically an architecture of molecules. Your DNA basically codes for the code for how to build you, which is really basically an assortment of organic compounds. The scaffolding of your body, whether it's proteins or DNA or lipids or nucleic acids, is based upon carbon. And there's no it's not a coincidence why carbon is the molecule of life. Carbon is tetravalent. That's a fancy word, which means that it forms 4 bonds. As we discussed in chapter 2, carbon has 4 valence electrons. In the second shell, it wants to have 8. So it forms 4 bonds, which is, in terms of stable molecules, that's about the most you can make. If you had 3 valence electrons, you don't usually form 5 bonds. Usually you just get rid of those 3 electrons. This little chart here, so carbon can do lots of cool things with those 4 bonds. It can form lots of different shapes and geometries of molecules. You don't need to have these memorized. It's just a good thing to note. So just to remind you, the 4 molecules of life that make up 96% of you, hydrogen, oxygen, nitrogen, carbon. Hydrogen forms 1 bond. It has 1 valence electron, wants 2. Oxygen forms 2 bonds. It has 6 valence electrons, so it wants 2 more. Nitrogen forms 3 bonds. It has 5 valence electrons, so it wants 3 more. And carbon forms 4 bonds. So memorize this. Hydrogen 1, oxygen 2, nitrogen 3, carbon 4. Okay, so I want to do a really quick, if the AP exam or if a question gave you a chemical formula and wanted you to be able to draw it, this isn't a huge thing to be able to do, but you need to for sure be able to recognize ones that are drawn incorrectly. So carbon dioxide, if I just said draw carbon dioxide, you always usually begin with the carbon, so just put a C in the middle. C's got to have 4 bonds. There's only 2 oxygens, so each of those bonds is a double bond. Each oxygen's got 2 bonds, the carbon's got 4. CO2 is a linear molecule, it's a straight line because double bonds the double bonds are pretty stiff bonds. CH4 is methane. This one's pretty easy. You draw a C in the middle, draw 4 H's, each H has 1 bond. The way this is drawn, it looks pretty flat. It's actually a tetrahedron, this geometry right here. That's not that important that you know that, but when you draw it, usually you just draw it flat. Urea is a component in urine. So there's a carbon and an oxygen, then there's 2 amino groups. That's what this notation means. There's 2 NH2 groups. So if you had to draw this, draw the C first, put the C in the middle. C has got to have 4 bonds. You could play around with trying different ways of drawing this, but the only way that actually works when you follow all the rules is the way I have it drawn here. A C double bond O with 2 single bonds to nitrogen. Then each nitrogen has 2 single bonds to hydrogen, so it has 3 bonds total. So all of the bonding rules are satisfied. The last one I want to look at is glucose, C6H12O6. This one, you know, back in the old school AP Bio days when I took it, you really had to have this memorized to be able to draw it from memory. That's not something that you need to be able to do now. But if you can draw it from memory, it means that you know all the rules and that you can apply this. So begin with the 6 carbons. Notice glucose is based on a 6-membered ring here in the middle. The top right atom is not a carbon, it's an oxygen. And the 6th carbon comes up here to the top left. So 1, 2, 3, 4, 5, 6 carbons. Once you've got sort of the carbon skeleton done, you sort of start putting on the other atoms. These groups, these OH groups, so like this is an oxygen bound to a hydrogen. You could draw a bond between them. Usually you don't. You just kind of shorten it as an OH group, but just recognize this is a carbon with a single bond to an oxygen with a single bond to a hydrogen. So the carbon's got 4 bonds in this case. The oxygen, this one has 2 bonds, and the hydrogen has 1 bond. So this carbon coming out of the plane has an OH group and then 2 hydrogens. The other carbons each have an H group and an OH group. So H group, OH group, H group, OH group, you see the idea. One thing to note, there's the idea of what's called free rotation, where the bonds can sort of rotate. This carbon, the OH group and the H group can be in either order. You could have the OH group pointed down if you wanted to. Why that is, I'll tell you in a minute. But the other ones, you can't. Like this OH group never points upward. It's fixed in that position. Another thing to note, so notice on the left side of the molecule, it doesn't say OH, it says HO. So on the right-hand side, you have, we kind of, in class we joke, we have O's. The left-hand side, we have H's. It's a biology joke, so don't get mad at me. Actually, it's more of a chemistry joke. So the reason is because you have connectivity, right? The bonds go carbon to oxygen to hydrogen. So if you're on the left-hand side, it's like a mirror image. You go from carbon to oxygen to hydrogen, which means if you write it out as a HO versus an O. So if you had this written like OH to the carbon, you'd be having 2 bonds to hydrogen. So the connectivity is always a carbon to an oxygen to a hydrogen. There is one, if you count the atoms, there's one atom missing the way that I have it drawn, and I did this on purpose actually. There's one atom that's not filling its bond rules, and that's this carbon right here. This carbon only has 3 bonds, right? So did I make a mistake? Well, no, the 4th bond is to a hydrogen. If you count the hydrogens, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, only have 11 drawn. So the 12th one goes right here. The sort of notation, the way you draw these is you don't have to show every single hydrogen. If there's a hydrogen not shown, you can just assume that it's hydrogen. So the 4th bond that's missing here, I didn't make a mistake. You don't have to show it. So actually, like this hydrogen, you could just not show. So carbons that are missing bonds, you just assume are hydrogens, and it's just so that the molecule doesn't get really messy when you're drawing it. These are, I think this is from your textbook. So one thing I want to point out, so notice, and this is going to be important later, there's 2 forms of glucose. There's the alpha form, and there's the beta form. And the difference is, like I said, remember how I said this OH group could be pointed down? So here it's pointed down, here it's pointed up. The alpha and the beta, that's the difference. And the deal is that with glucose, when you dissolve it in water, glucose can linearize, which means the ring can break open. This bond right here, the O to the C bond, that bond can be broken. And if you did that, and spread it out, you would have what's called the linear form. It would be basically a straight line. And glucose can go back and forth. It can linearize, it can become a ring, it can go back and forth if it's in water. So based upon how this bond forms, this O could be pointed up or it could be pointed down. So once you're in a ring, it can't switch. But because it can go from a ring to a line to a ring, it can switch in solution. And this is going to be very important later. I'm not just saying this just for giggles. This is an important concept that we'll see an application of later, probably in video 2 of chapter 3. So the alpha form, the OH points down. The beta form, the OH points up. And notice the way this is drawn is correct. On the left-hand side, you have HO groups, you have HOs. Some textbooks don't do it right. Some textbooks would just say OH, but that's incorrect connectivity. So I don't like that. But this, the way it's drawn here, is correct. Okay, this just shows 2 other organic molecules. Estradiol is a form of estrogen, testosterone. These are both sex hormones. So notice, I have it's 4 rings that are fused together. Wherever you see a corner, that represents a carbon. So like this atom right here, that's a carbon with a bond to carbon, a bond to carbon. So 2 of the bonds to this carbon are not shown. So they would be hydrogens. So sample question, how many hydrogens are bound to this carbon right here, right? That carbon there, you know it's a carbon because it's a corner. There's 1 hydrogen bound there that's not shown. How about to this one? So this is a carbon with a double bond to another carbon, single bond to another carbon. So it's got 3 bonds the way it's drawn. So it would have 1 bond to a hydrogen that's not shown. This carbon right here has no hydrogens because it's got single bond to carbon, double bond to oxygen. So it's full. Yeah, you don't need to know how to draw these molecules, but again, using the rules to pick out the correct molecule or things that are wrong, that's something you should be able to do. It's also worth noting, estrogen and testosterone are very similar looking molecules. The only difference is this methyl group here, there's some double bonds in this ring different, and then whether it's a double bond or a single bond to this oxygen, and here it's an OH group. That's the only difference. Okay, so like I said, it's no coincidence that carbon has the foundational molecule in biology. Carbon does lots of things that are useful. One is it's tetravalent, which we've already discussed. Carbon can form chains that are, gosh, thousands of carbons long. It can form chains that can branch. It can form rings. You can have chains with rings in it. You can have chains with branches and rings. You can have all kinds of cool scaffolding, cool molecular architecture with carbon. Just really quick, so these pictures I'm going to show you are just ones from this slide. So these 2 molecules, this is called butane, this is called 2-methylpropane or isobutane. The real difference here is whether, so here I have a chain, here I have like a chain with a branch. Both these have 4 carbons, right? It's just the arrangement is different. They both have 10 hydrogens, right? 1, 2, 3, 4, 5, 6, 7. Yeah, they both have, so the formulas are the same. It's just, do you have the structure of what's bound to what the same? And the answer is you don't. They're called isomers. We'll get to that in a minute. Where the double bond is, so this molecule is called 1-butene or 2-butene. The -ene suffix means there's a double bond. So here the double bonds at the end. Here it's in the middle. So same formula, different structures. Notice like the carbons with the double bond have fewer hydrogens. So this carbon right here has 3 hydrogens. This one only has 1 because it has the double bond. So one like theme to have in your mind is when you add double bonds to a molecule, you have to lose hydrogens because carbon, again, can't have more than 4 bonds. Presence of rings, this is cyclohexane. This ring is called benzene. Benzene's a cool molecule. Notice here I added 3 double bonds. And when you add double bonds in the ring, you lose hydrogens. The word hydrocarbons, so the word gives it away. They're just molecules that consist of hydrogen and carbon. That's it. So oils, like hydrocarbons are the gasoline, oils that we use for fuel. They're just long chains of carbons with a bunch of hydrogens attached. They release lots of energy. Fats, when your body stores excess food or excess energy as fat, that's a hydrocarbon, right? Because they store lots of energy very efficiently. So if you burn fuel or if you burn off calories, you give off the energy because you're breaking the bonds. Isomers, like I said, are just compounds that have the same formula but different structure. So going back, like the butene or 1 or 2-butene, it's just based upon where that double bond is. Okay, so functional groups. So functional groups, there's going to be a bunch of them that we're going to go through. The carbon skeletons are great They're scaffolding. They're what supports the molecule. The functional groups really tell you what the molecule is going to do. I had a professor in college at Furman who called functional groups like shrubbery, because you build a house, that's the house. You don't really change the structure of the house, but the shrubs you have in front you can change. You can move them around, you can plant them, you can get rid of them. He called functional groups molecular shrubbery, because that's what you put around the structure, and that's what kind of gives the molecule its flavor. If it's going to be acidic or basic or polar or non-polar, things like that. And you can change them with chemical reactions. So we're going to go through 7 of these. This seems like a lot, and it is, but the nice thing is once you know what the functional groups do, like you know what the molecule does, and the themes just kind of repeat. So this is from the book. This just reviews them all. So let's go through these one at a time. So a hydroxyl group is an OH group. So first off, this notation R stands for the rest of the molecule. So like in my example here, ethanol, which is the substance found in alcoholic beverages. So here's my OH group, right? The rest of the molecule is this part right here, these 2 carbons and these 5 hydrogens. That's what R stands for. So R is just a notation for what's for the rest of the molecule. The first bond to R has to be a bond to carbon. So like this bond right here in this notation is this bond when it's pictured. Hydroxyl groups, OH groups, are called alcohol groups. Remember, OH is very polar. We did this in chapter 2. So things that have hydroxyl groups mixed with water. So ethanol will mix with water, no problem. They're polar, so they mix with water. So if I want to make a molecule polar and make it more polar, you add OH groups. And you can add them, you can take them off. OH groups are molecules that have these tend to end in the suffix "-ol", like ethanol, propanol, things like that. Okay, cool. That's hydroxyl groups. Carbonyl groups are C double bond O. So notice here I have a carbonyl group. It's just a C with a double bond to an O. Aldehydes and ketones. Look at the picture. So propanol in this picture, the C double bond O is at the end. So the last bond is just to a hydrogen. So like this carbon right here has a bond to another carbon and then a bond to a hydrogen. That's called an aldehyde because it's at the end. A ketone, like acetone, which is in fingernail polish remover, the carbonyl group is in the middle. So this carbon, both bonds to the side are 2 carbons. So a ketone has 2 bonds to carbon. An aldehyde has a bond to carbon and a bond to hydrogen. Yeah, okay, cool. Carboxyl groups are carboxylic acid groups. They're the same thing. It looks like COO. The connectivity is not COOH. It's a carbon with a double bond to an oxygen and then a single bond to an OH group. They're called carboxylic acids because they tend to make the molecule acidic. If you look at the picture, this hydrogen right here, this hydrogen, this is the kid that gets bullied at lunch because that oxygen and that oxygen are both very strong electronegatively speaking, and they're going to pull the electron away from hydrogen. So much so that it might just get kicked off. And if it gets kicked off altogether, then that's an acid, right? Because you gave off an H+ group. One little aside, well, let's do a quick inquiry question. So here I have acetic acid, which is vinegar. Look at the formula. There's a double-headed arrow here, right? So both forms of this molecule can exist. Here I have one where the OH is intact, and here I have one where the H+ got kicked off, and I'm left with O with a negative because it has that extra electron. So say I have a beaker of vinegar. I can sort of play with which form is most predominant, which one's most available. And I can do that by playing with the pH, okay? So common AP Bio question, right? If I had this in a solution that was very acidic, say it was a pH of 4, say this is A, this is B, which form would be predominant if this was now I know vinegar is a weak acid, but say that I added some hydrochloric acid to it to make it super acidic. Which form of the molecule would be more predominant, choice A or choice B, if it's in an acid? Think for a second. The answer is choice A, all right? Think to yourself, am I forcing this hydrogen on the molecule, or am I encouraging it to leave? And if I'm in a solution that's very, very acidic, you have tons of H plus. So that's like pushing the reaction this way, pushing this H plus on the molecule. Same question, it's in a pH of 10, which form is most predominant? It's going to be choice B here, because if I have less if I have less H plus, it's more likely it's going to get kicked off. All right. So if I want to favor the forward reaction going this way, you make the solution more basic. If I want to favor the reverse reaction going this way, you make the solution more acidic. Okay. That kind of inquiry thinking is something you should be able to do. Okay, cool. Moving on. And amino groups. So amino groups are NH2. They're called amines. They contain nitrogen. Notice, so amino groups tend to be bases or tend to be basic, because if you look at the picture, you can force an additional H plus onto an amino group. It forms NH3 plus. And if a molecule accepts H plus, that makes it a base, right? So adding amino groups tends to make the molecule basic. Sulfhydryl groups, these are not that important. There's a couple places where they pop up later in the class. They contain sulfur. They're also called thiols. It's just an SH group. Sulfur bound to R and then bound to a hydrogen. Phosphate groups are groups that have a phosphorus atom and 4 oxygens. They're super duper negatively charged. You know, DNA contains phosphate groups, right? Notice the way this is drawn here. This would have a -2 charge because there's 2 oxygens or 2 oxygens with only a single bond, which makes the oxygen atom negatively charged. Okay. Methyl groups is the easiest one. Methyl groups are just CH3. Remember, methane was CH4. So a methyl group, which is a CH3. Carbon with 3 hydrogens, the 4th bond is to R. You can throw methyl groups on DNA, on proteins, kind of willy-nilly. The verb is to methylate if you add methyl groups. Okay, so going back real quick. So in the textbook has this diagram. You need to know this diagram. Like, you need to know not so much examples, but what the functional groups are, what they do, acids, bases, charges, positive, negative, things like that, in terms of the 7 functional groups. Okay, cool. Let's see. So ATP. So in a later chapter, there was a whole section on ATP in chapter 6, maybe, I forget. So we're going to come back to this in a lot more detail. You already know from freshman bio that ATP stands for adenosine triphosphate. It's the primary energy carrier of a cell. It's 3 phosphate groups, hence triphosphate, with a molecule of adenosine. What I want to go ahead and talk about now, so this is 4 negative charges, like really close to each other. This molecule is not particularly stable. ATP does not like having these 3 phosphate groups bound together. It's like a molecule that's very hyperactive. Think of it like you took a spring and you coiled the spring. Coiling the spring took energy, right? If I let the coil go, it's gonna uncoil itself. Coiling it takes energy. Uncoiling it doesn't take energy, right? It gives off energy. So ATP took energy to make, and this molecule would love to get rid of this 3rd phosphate group. It would love just to give it to any other molecule that would take it. Doing that would lower its energy, and if you do that, you make ADP. So here's ATP. You need some water. This is high energy. If you kick off that 3rd phosphate group, it's called an inorganic phosphate now. It's a P with a subscript I. What's left is ADP, adenosine diphosphate, and you gave off energy. Whatever molecule you gave that phosphate group to, you phosphorylated it. That's the verb, to phosphorylate. And you transfer energy to that molecule. So ATP is a super important molecule. It takes energy to make it. You give off energy to break it down. And again, we're going to come back to ATP in more detail later. Okay, so we're nearing the end of part 1, I promise. So macromolecules, fancy word for big molecules. There's 4 main macromolecules in life. Carbohydrates, proteins, lipids, and nucleic acids. Part 2 of this video is going to go into the 4 types, and there's a lot to go into. So basically, it's like taking Legos and putting them together, or taking cars of a train, linking them together. The polymer is what you call the whole thing, like DNA or your Lego creation. They're made of small building blocks called monomers, right? So you link monomers together to form polymers. The monomer of DNA is called a nucleotide. The monomer of proteins are called amino acids. We're going to go through all four in LSA in part 2. What I do want to go ahead and do now is talk about the kinds of reactions that make polymers or that break them down. And this is super important. This is something that you need to know very well. So if I want to link two monomers together, the reaction's called a dehydration, or some textbooks call them condensation reactions. And you call them that because they give off water, right? So say I have two monomers right here. Here's monomer 1, here's monomer 2, and I want to form a bond. So this number 3, this represents something with carbon, and this little gray dot here is something with carbon, and I want to form a bond between those two carbons. Right? Well, right now this has an H, this has an OH, or here it shows an HO. I'm going to bind this 3 to this one, and in doing so, I'm going to give off water. This H and this OH is going to be combined to form water. So you formed a new bond here, and you gave off water. Hence the word dehydration or condensation. I forget which term your textbook uses, but you should, I think your textbook might use dehydration synthesis, I forget. They both mean the same thing. So you form a bond, you make a polymer by giving off water, okay? Sample question. Say that I had 100 amino acids, which is the building block of proteins. I had 100 amino acids, and I'm going to link all of them together into one long chain of 100 amino acids. First question, how many bonds do you have to make to link those 100 amino acids together? How many bonds would you have to build? Okay, think about it for a second. If you want to, pause the video. You'd have to build 99 bonds because that first bond links 2 of them, right? So if you link together 100 amino acids, how many water molecules would you make? You would make 99, okay? Go even further. If you, actually, let's do a different number. Say that you link 10 monomers together, right? You would make, 9 bonds then. So what if I said, how many hydrogen atoms, not how many waters, how many hydrogen atoms would you be giving off if you link 10 monomers together? Well, linking 10 monomers would give off 9 waters, which is 18 hydrogens, right? Because it's H2O, okay? Does that make sense? These kind of like very simple math problems the AP exam might have you do. So one more, you link 30 molecules together. That's going to give off 29 waters, and that would be 29 oxygens, and 29 times 2, I'm not doing math in my head right now because I know I'll get it wrong. Do the math yourself. It's double because there's 2 hydrogens in every water. But it would be 29 times 2 hydrogen atoms. Okay, cool. Now, this next one is just the same thing in reverse. A hydrolysis is breaking that bond. The word hydrolysis, hydro means water, lysis means to break, right? So it's literally breaking a bond by shoving in a water molecule. So the polymer that I made, this bond here that I made in the dehydration reaction, I'm going to break it. And in breaking it, I'm going to shove in a water molecule, okay? When I was in school, I always forgot these 2 terms. I couldn't remember them, or I would get them confused. But the word hydrolysis, the word means water breaking. So you're breaking a bond using water. So if you remember what a hydrolysis reaction is, the other one, dehydration or condensation, it's just the opposite. So same question or sample question. I want to break 100 bonds. How many water molecules do I need? I need 99. Because when you break that first bond, now you now have 2 pieces. If I want to break 10 bonds, you need 9 waters. Okay, cool. So we're going to stop there in part 1. In part 2, we will talk about the 4 categories of macromolecule. And that's some pretty thick stuff. So we'll come back to that in part 2. All right, hope that was helpful. See y'all later.